Thank you very much once again. Let me take this opportunity to welcome you back at this one aristocratic channel of ours. And then today we've got what we call an important measure, which is a paper that was written last year, September 2023, paper 2. This is pertaining life sciences paper 2. And then let us ensure that we use this effectively. We use this into measure because of now we've got to write paper 2, and then today we are starting at section A. Ensure that you are sharing to those who are concerned and ensure that you are hitting the subscription button. 3 pieces, so ensure that you are hitting the subscription button. As we know that under section A, they are saying various following question. Time one, answer A. Point 10 is number D. So in question 1, we are there to find out in which phase do the spindle fibers start to form. And then we know that when they form the spindle fibers there, they are handled by the central zone there. And then we are saying that it happens on the prophase 1. Remember those stages. Ensure that you are referring back to the content that we have been giving. In 1.1.2, they say the following includes some of the features that are studied under the evolution part. And this is the last chapter that we have done. And we have talked about the bipedalism because of now they can be able to work with 2 feet and hand object with the 2 hands. So we are saying that they increase the chances of the genitals part and which it is used to attract the other genders. We talk of the dentition. We know that for us as the Homo sapiens, our dentition in terms of we having the smaller canines, but other primates, they've got the bigger dentition. And then we are saying the food that they eat, it is because of them. And then we talk of the brow ridges, which we find the difference between other primates and humans, and the stereoscopic vision. And then the question, it says, which of the key features separate the human lion and from the African ape? So we are saying that it would be number 1, number 2, and number 3. 1, 2, and 3. So we know that it is number D because of we are having these different features between us as Homo sapiens and African apes. And then if ever we are going on again, we are to see that in this instinct we are having 1.1.3. We are saying in the section of the mRNA, the mitochondrial RNA synthesis. Just bear in mind, we are saying that has the following sequence of the bases that codes. So we are in chapter 1 in this instinct for the different amino acids. Remember that we are giving us of the amino acids that are there. We are saying that the synthosine, 3-synthosine, and the synthosine guanine uracil, and the use for uracil adenine and adenine. We are saying which one of the following of amino acids coded for by this section. So in this instinct, we are saying that this it would be number, it is indeed number B, which is it has the 3 amino acids there. And then if we are moving on again, we are to see that this GGG, the guanine will go with the synthosine that is there, and this GC, and then this one GCA, it would go with the the GCA that is here. It will go with the synthosine, the guanine, and the uracil. And this one, the AUU, it will go with the uracil, the adenine, 2 adenines. And we know that there are 3 of these amino acids that we are talking about. And remember that amino acids, they are building blocks, or they are known as the monomers of the protein. Right, if ever we are going on again, have the courage to watch this video up until the end. And then we are having now one of the anticipation in question 1.1.4, whereby we are saying in a family of 4 children, each child has a different blood group, and then they derive from the same family. And then we are saying the genotypes. What is the genotype? It is the genes that they inherit, that they will have the physical appearance, which is called the phenotype in this instinct of the parents. It must be an A superscript B and the small I, which is a recessive. So we are saying in this instinct is number A. And then if ever we are moving on again, we are to see which one of the following monohybrid crosses remains now 1, where complete dominance applies, will result in the phenotypic ratio of 3:1. We are saying that it would happen when both parents are heterozygous. Heterozygous, it means now one is dominant and the one is recessive. So we are saying that is heterozygous. And then if ever we are moving on again, we are to see that we would go to question 1.1.6 and 1.1.7. So we are saying here, are based on the timeline that are situated here below, showing the possible evolution of some humanids. We are saying we are giving this table, and then on our left hand side, this is time in millions of years ago, and then we are to see that it starts for the oldest time and to the least time. So we are saying that these Ardipithecus ramidas, they existed between 5 to 4 millions of years ago, and then the Australopithecus afarensis, they derive from 4 to 3 million, 3 to 4 million of years ago. And then we talk about the Homo habilis, and then we are saying it's 2 million of years ago. And then we can talk of the Homo erectus, which they derive from 2 to 1.5 to 2 million of years ago. And then we've got the Homo sapiens, which is 0 to maybe in 500,000 years ago. So we are saying that according to this feature, the question in 1.6, it asks us very well to say which now species inhabited the earth for the longest time and how many years ago did the species appear. So we are saying that this is Homo erectus in this instinct, which it is now on earth for 2 million of years ago. And then in this instinct, we are moving on again to see the exact answers there, which we can actually see. And then if ever we are moving on again, we are to see that the longest time in this instinct, that is a mistake, but we are saying the correct answer there. It is this of the 5 million of years ago, which is number D. Sorry for the error there. And then if ever we are moving on again, we are to see that we've got a question to say how many genera are represented on the timeline above. And then we can see that there are 3 genera in terms of the generation that they have been interpreted there. And then if ever we are moving on again, we are to see that we are having the other questions which can derive there. And then we are saying the diagram below represents different phases of meiosis. And then you can see that these are different phases, and these are the chromosomes. And then we've got the centromere, and then they are held by spindle fibers. So you are to identify these particular phases. And then they say here, identify the correct phase in diagram 2. So here it is diagram 2. You can see that it is metaphase 1, whereby now these spindle fibers, they are holding these chromosomes on the upper poles, the north and the south poles. If we are moving on again, we have to see that we've got one of the question in 1.1.9, and they say if a DNA molecule contains 20,000 nitrogenous base, of which 20 are cytosine, how many adenine molecules will be presented? And then we are saying that is 6,000, because of that is the 20%, is 20% over 100 times 20,000. Then you get your 6,000. And then we are saying that is the correct answer, because of it is a 20% there of 6,000 over 100. That it must be there times by 20,000. So that is the correct now calculations in that instinct. And then if ever we are moving on again, we are to see that we are having some of the questions which need our attention, this instinct. And then we are saying when we are moving on, let us ensure that we are watching up until the end of our video. And then if ever we are moving on, we move to 1.2.1. But here we are giving a statement in 1.2 to say give the correct term, biological term for each of the following description. Write only the question number in 1.2.1 and up until to 1.2.9. In 1.2.1, we say that the mode of locomotion where the animal uses the 4 limbs is quadrupedalism. And then the bond that is formed between amino acids, it is called the peptide. And then if ever we are moving on, the part of the skull that houses the brain is called the cranium. And then in 1.2.4, a change in sequence of nitrogenous base, it is called the gene mutation. And then in 1.2.5, a disorder due to the presence of the extra chromosome on the 21st pair in the soma called the trisomy. And if we are moving on again, we have to see that we've got the gene evidence is used to support out of Africa hypothesis, and that is mtDNA, the mitochondrial DNA. And if we are moving on, we are to see that we've got 1.2.7, whereby the question it says the process by which the genetically identical organisms are formed using the biotechnology that is known as cloning, and the type of dominance in which both alleles are expressed in the phenotype that is co-dominance, or it is partially a dominance. If ever we are moving on again, we are to see in 1.2.9, we are saying similar structure that are inherited from a common ancestor and modified for different functions. That is homologous structure there. It is homologous structure. And then if we are moving on again, we are to see that we've got the other question that follows, and it needs our attention in this regards. If ever we are moving on in 1.3, we are to see that indicate whether each part of the statement in column 1, and here is column 1, to A, B only, both A and B, or none of the items in column 2. Write only the letter A only, B only, A and B, or none next to the question number that is given. And then in 1.3, without wasting any time, we are saying that the gene of a person with hemophilia, the blood hemophilia, we are saying that this is correct. The X superscript H and the XX with the superscript H, we are saying the presence of the hemophilia is there. And then we are saying that is both. And then we are saying in 1.3.2, the variation in human height, it is called a continuous variation. It is not a discontinuous, so therefore it's A only. If we are moving on in 1.1.3, it is involved in the discovery of the DNA molecule. And then we can see that none of these did it molecule, therefore we write none. Okay. If we are moving on again, we are to see that we still have some important aspects that we need to consider in this instant. And therefore we are saying in 1.4, ladies said there, each time they gave a birth to a baby boy in the hospital, the babies were accidentally swapped. The diagram below shows the technique used in paternity testing and the result of each parent and their baby boys. And then we can see that we are having 3 parents. Is for Mr. and Mrs. Smith, Mr. and Mrs. Stevens, and Mr. and Mrs. Jones. And here are the 3 kids, these babies. So we are given in terms of matching them. We are trying to find now the swapping of the baby. And then according to this, in 1.4.1, they say identify the technique shown above. So this technique that has got these bars, you must know that is DNA profiling. In 1.4.2, which baby 1, 2, or 3 belongs to Smith? So we are saying it's baby 3. Let us check. So how do we check? We go to Smith, and then we can check there for Smith. So we are saying this one for Mr. Smith is there, and then the baby 3 has got a code there. And then we are saying this one for Mrs. Smith, the baby is having the code there. And this one, it is for Mr. Smith, the baby is having a code. And then this one, it is for Mrs. Smith, and the baby is having the code there. So they are deriving from that particular parent. That is how you see the matching of those graphs. And then if ever we can go on again to the question, you can see there, it says in 1.4.3, state 2 other uses of techniques named in question 1.4.1. So we are saying we use DNA profiling. Number 1, to identify suspect in a crime. Number 2, to trace a missing person. Number 3, identifying genetic disorder. Number 4, identify a dead person. So you get your 2 marks there. And then if ever we are moving on again, we are to see that we still have some other important aspect that needs our attention. And in this regards, that is 1.5. And then in 1.5 here, you are to see that an investigation was carried out to determine the effect of the antibiotic on the bacterial resistant. So we are saying that we want to see the bacteria being resistant to a person. And then we are saying the diagram below shows the effect of the antibiotics on the bacterial population. So this graph number 1, this is the original bacterial population before the treatment with antibiotic has been given. And then we can see in the features, these are population which are resistant to bacteria, which are 1, 2, 3. And the rest, they are non-resistant to bacteria. 1, 2, 3, 4, 5, 6, 7, 8. There are 8. And then if ever we are moving on, we are saying that the bacteria population is treated with antibiotics there. And then when it is treated, you can see that the antibiotics are being placed here. And when they are placed, you can see that in number 3, the bacteria population after the treated with the antibiotic, you can see that this is the key element to see that they have killed these bacteria here. The antibiotics have killed the bacteria, but there's only one that is left here. And if ever we are going again, we would see that there will be a formation of the new population whereby the new bacterial population, they are resistant, they are there, they are resistant because of the effect of antibiotics. And then in 1.5.1, we are given to say, give the dependent variable. Dependent variable, it is something that is depending on something to happen. So we are saying that is bacterial resistant. And number 2, identify the independent variable is the effect of the antibiotics, not antigen. So it is antibiotics, biotics. So that is antibiotics. And then we are saying there, name the type of the evolution that occurs in this bacterial population. We are saying that is natural selection for NS and punctuated equilibrium for PE. NS, natural selection, I repeat, and PE, punctuated equilibrium. If we are moving on again, we can see in 1.5.3, we are asked what is the change in the environment that brings about the change in the bacterial population. We are saying that this is because of the effect of the antibiotics. You get your mark? And then if ever we are moving on, we are to see that which variation can be found in greater proportion in the final population. We can find that the variation is resistant. What differs is the resistance that the population they have because of the effect of the antigen and antibiotic. If ever we are moving on again, we can see that we are asked in 1.1.5 to say what is the percentage of bacteria in the original population was resistance to antibiotics, and then we say 25% based on the calculation that we are given there. So we are saying that it will be 20 of, we are saying 1, 2, 3, and 3 over, so we are saying 3 over 4, 3 over 12, because of there are 12 of these times 100, then that we are saying is 25%. And then if ever we are moving on again, we are to see that we are having now some of the important thing, a lesson to ensure that we understand it. We can also refer to the previous modules. We are saying that a genetic disorder is caused by a recessive allele. So we are saying which is A, and then we are saying an individual with the disorder is described as affected. And then if ever we are moving on, we are saying that, and an individual without it is described as unaffected. The affected individual is indicated by the black circle or the square. The diagram or the pedigree diagram below shows the inheritance of the disorder in the family tree. So we are saying that that is them. So we can see these are the parents, A and B are the parents which they later on have children, and their children, you can see there are 4 of them, and this one is affected, and this one it is also affected. So the 2 here, they have children, and then which is now these 2 that are there, which are unaffected. So they say here, how many of each of the following are presented in the diagram. We want now males that are in the diagram. I'll show you. And we want the generation, and we want the children of A and B. So we want the males and the generation. So if ever we are moving on again, we can see that we are having the calculation of the males in this instinct. We are saying that we've got and 3 of these. So it is 5, 3, and 3. So how do we get that? I want us to understand this concept. So we are saying that in males, we are having 5, and then generation 3, and then we are saying that that is 3. And then how can we see that we are having 5 males? By calculating now these. So we are saying this, they represent male, that is 1 male, 2, 3, 4, and 5. So we've got 5 males in this, and then we've got 3 generation. How do we say that we have 3 generation? This is generation number 1, this is generation number 2, and this is generation number 3. Okay, and then if ever we are moving on, they say now give here the children of the individuals A and B. So we are saying there are 3 of them. So we are saying A and B, they have 1, 2, and 3 children. So that is how we can see it. So we are saying this is 1 children, 2 here, and then this is 3. Why do we say that? Because of the fellow diagram that is coming on to this population here, or to this particular offspring that are there. If we can move on again, we have to see that we've got some other 2 questions. We say in 1.6.2, give the possible phenotype of the individual D. The phenotype is the physical appearance. The genotype is the genes that he carried. And then in phenotype in individual C. So we are saying that we want the genotype and the phenotype. So if ever we can look very well there, we are to see that the genotype there, it is A small, this dominant A, and this A, which is you are allocated to your mark. And then we are saying that the affected female there, you are giving to say this one, it is an affected female. How can you see that is an affected female? By the color now. By the color, remember that we have said that these squares which are black, they represent now the affected person. So the possible genotype of D, we have seen it, and then the phenotype there of number C. So we are saying this is number D, which can be AA, because of there is no presence of this disease. So the A, they are big there. And here we are saying that lastly, we are talking about this physical feature, of which now this is the physical phenotype of C. The phenotype of C, we go to the phenotype of C. So here it is C. So we can see that this black dot, it means is an affected, and this circle, it means that is a female. So is an affected female, the UF. Thank you very much for tuning on in this opportunity. Please tune on again for the section B, which is question 2 and question 3. The reason why we are breaking it, we want you to understand, not to put yourself under pressure when you are studying this. So let us break it here, and then let us ensure that we are hitting the subscription button, and ensure that we share to those who concerned, even on our social media platform. At MATRIC+NATED Specialist, thank you very much for your time.